Greetings and welcome to the ABCs of Cancer Risk. Understanding what risk means for BRCA mutation carriers. If you have not already done so, please close all of the programs on your computer. You may submit a question via the web at any time by typing them in the Ask a Question section on the left side of your screen. If anyone should require technical support during the conference, simply click the question mark icon on the top right corner of your screen. It is now my pleasure to introduce Pamela Klein, Outreach Manager for Vassar Center for BRCA. Please go ahead.
Good afternoon, and thank you for joining us for today's webinar. The Vassar Center for BRCA at Penn Medicine's Abramson Cancer Center is the only center in the world solely devoted to BRCA-related cancers. The Vassar Center supports cancer care and BRCA research at every stage, including communication, outreach and risk assessment, prevention, early detection, treatment, clinical trials, and survivorship. Dr. Edward and Merry Prostic have generously established this webinar as part of the Elizabeth Prostic Memorial Outreach Program in memory of their daughter. The following video is a tribute to Elizabeth, her incredible life, and her dream of creating communities that increase awareness about BRCA and metastatic cancers. The Vassar Center is proud to be a part of this mission by providing life-saving information and hope to individuals and families worldwide. Please begin the video.
Tomorrow, bet your bottom dollar that tomorrow there'll be sun.
Lizzie Prostic was an amazing young woman who believed in the promise of tomorrow. And though her tomorrows were cut short, we can celebrate and sustain her optimism. Lizzie lived large. She loved the stage and brought it to life with her voice and her energy as a talented child and as she grew into a woman. Her world was full of loving friends and family, and success in class, in sports, with girls, and with boys. Sophomore year of college, Lizzie met Michael Lundblad, a liberal and an English major. He got a preview of her leanings when he saw her quizzing a friend about senators. But she learned to love camping. He learned about couture. And political opposites, Michael and Lizzie spent 10 years together, marrying their minds and their passions. Michael loved her boldness and her softness, her interest in so many different types of people, and ability to remember the small details of their lives. The time she made to talk with her grandmother, her pearls, her red shoes that said she was not afraid to be noticed. She was not afraid. Lizzie's intellect and drive were rewarded with exciting opportunities. She blazed through the University of Pennsylvania and into the halls of government, determined to win in every venue and for every cause that was important to her. Her work for senators and on committees quickly made her a Washington insider. She rose to Chief Privacy Officer and Senior Advisor to the Secretary of Commerce before being lured away to the private sector. Not yet a lawyer, she was named Managing Director at Sonnenschein, Nath, and Rosenthal in Washington, D.C., and was completing her law degree at night at George Washington University when she died. When Lizzie was awarded a posthumous degree at the law school graduation, the entire audience stood in tribute to her spirit. Lizzie was 31 when she was diagnosed with stage 4 metastatic breast cancer, and her daughter Harper was 4 months old. Michael did research. Lizzie didn't want to know the odds, expecting to beat them. But the cruel reality was that although breast cancer is generally curable, metastatic breast cancer is not. And they found little support to help them deal with the psychological and emotional realities they had to face. In her last months, Lizzie and her family began to envision an online support network that would show patients with metastatic disease that regardless of how long they have to live, they have the ability to control how they live. This was Lizzie's final cause, and one which will be honored. Lizzie wanted to see her daughter's first birthday. She didn't make it, but she lives on in the people who loved her and through grants such as this one being made in her name. And in truth, none of us knows how long we'll live, so we all should listen to Lizzie and make the most of every tomorrow. Live large, live strong, and wear red shoes.
Today's program will discuss our current understanding and definitions related to cancer risks in BRCA carriers, as well as research that seeks to refine those risks. You may type questions into the Q&A box on your screen. We will try to answer as many questions as possible at the end of the presentation and during the presentation. Today's program will be archived and available for on-demand viewing. I'd like to introduce our speaker, Dr. Susan Domchek. Dr. Domchek is a board-certified medical oncologist, Vassar Professor in Oncology at the Perelman School of Medicine at the University of Pennsylvania, Executive Director of the Vassar Center for BRCA, Director of the Mary and Robert MacDonald Cancer Risk Evaluation Center at the Abramson Cancer Center, and a Senior Fellow at the Leonard Davis Institute of Health Economics. Dr. Domchek has committed herself to pursuing research related to genetic susceptibility to breast and ovarian cancer, particularly with regard to risk assessment, screening, prevention, and treatment. She is especially interested in the development of targeted therapeutics in this population, as well as the long-term impact of risk-reducing interventions. Dr. Domchek, the floor is yours.
Great. Thank you so much for allowing me the opportunity to speak with you today about this important topic. We will be getting into some of the details about risk, and we will be using some bigger terms, so feel free to interrupt or to ask questions as we go on. Just want to make sure, Pam, if you can make sure those slides are there. I'm not seeing them pop up. There we go. I think I figured it out. Sorry about that, guys. There we go. All right. So the first thing I'm going to do is just to describe some of the terminology. When we talk about risk, this only means the probability that an event will occur. And we always like to start this conversation by saying that having a BRCA1 or 2 mutation or having any kind of a genetic susceptibility, the vast majority of the time, it does not mean that you will get a disease. It means that you're at significantly increased risk for the disease. And so it really is important to know that there are people who have mutations that do not end up having a disease over the course of their lifetime. So we use different terms to describe risk. One that you may commonly see is something called relative risk. And this is a proportional measurement. It means the risk of an event happening in one group, such as BRCA1 carriers, compared to the risk of the same event happening in another group, which is the control group, and that's generally the general population that we refer to. When a relative risk number is higher than 1, that means that there's an increased risk. When the number is lower than 1, there's a decreased risk. So depending on the particular feature you're looking at, you can have relative risks that are both higher or lower than 1. The second way that we define risk is by absolute risk, and this is the chance that an event will occur expressed as a percentage. And it is really looking at, over the course of time, the ratio of people that might have an event over all that could possibly have that event. So to put this in sort of practical terms and to show you what we mean by this, you can look at the relative risk of a man with a BRCA2 mutation having a development of breast cancer, and that risk increase is sort of a 10 to 50-fold increase. And if you look at it this way, the absolute risk in the general population is sort of 1 to 5%, compared in the BRCA carriers is 1 to 5% compared to the general population risk of 0.1%. So you might have a relative risk that's as high as 10 to 50, but you have an absolute risk that's in the 1 to 5% range. So it's really important when you're talking about risk to know whether we're talking about relative risks or absolute risks. In addition, particularly in the media, it's very common to express risk a little differently. So I wanted to point out a specific example. There are genetic susceptibilities that we call single nucleotide polymorphisms that increase the risk of breast cancer by just a tiny little bit. So a relative risk may be 1.2, which is a very small relative risk. However, frequently in the media, this might be expressed as a 20% increase in the development of breast cancer, which sounds a lot worse when you say it that way. In addition, if you take into account that the absolute risk of breast cancer for a woman in the general population is about 10% by age 70, a SNP, single nucleotide polymorphism, with a relative risk of 1.2 will only raise that risk to 12%. So you can see how if you say things differently, it feels very different. You can say a relative risk of 1.2, that sounds not bad. You can say that it's increasing risk by 20%, which sounds a little scary, or you can say that it changes risk from 10% to 12%. All of those are accurate, but they can feel very different when you're hearing that information. And yet there are more terminology associated with risk. There is something called cumulative risk, which is an incidence proportion. And this is a measure of disease frequency during a set period of time, which is usually expressed as a lifetime risk, so say until the age of 70. And it's the probability of an event occurring before a given time when all the individuals are at risk. And this actually gets into issues of the fact that when BRCA1 and 2 mutation carriers, when we're looking at risk, we often take out patients who've had something else happen to them, and that can impact whether we're talking about how a cumulative risk and an absolute risk may differ. Again, it doesn't matter so much. Those differences are low compared to this idea of relative and absolute risk. Another really critically important aspect that we don't talk nearly enough about is the issue of confidence intervals. So from a statistical standpoint, confidence intervals are the range of the expected result if the experiment were repeated many times. We all know that if you do the same thing many times, if you run a certain distance 6 times, you probably aren't going to get the exact same result 6 times in a row. And so we take sort of an average of that. Think about it that way, which is a confidence interval expresses the time you ran at the shortest, the fastest and slowest within that time interval, and it gives you a sense of the range. And it's really important to understand confidence intervals to determine how accurate the number is that's represented as risk. So if there's a very wide confidence interval, we're not so certain what the exact number is. If there's a very small confidence interval, we're much more certain in the fact that that may be a pretty accurate number to portray. Finally, the issue of hazard ratios comes up. Hazard ratios are a ratio of hazard rates, and they are different than relative risks because relative risks are cumulative, generally over the course of a lifetime, where hazard ratios represent a risk at a specific time point. Again, these are fairly subtle differences from a statistical standpoint, but if you can remember relative risk, confidence intervals, and absolute risks, then you're going to be doing pretty well. So with all that really mind-numbingly boring statistical definitions in mind, let's try to get at the issues of how that applies to BRCA1 and 2 mutations. So what is the risk of my risk of having a BRCA1 or 2 mutation? What are my lifetime risks of cancer if I have a mutation? And how do what we call age-specific risks factor in if I have such a mutation? We try to express risk in different ways because people do understand information differently, and visually this can be very helpful. So this is one example of that, which is in the general population, the risk of having a BRCA1 or 2 mutation is around 1 in 400. However, if you're of Ashkenazi Jewish descent, this is 1 in 40. And you can tell that if you do this pictorially, it really does get that point across. You can use percentages, you can use numbers like 1 in 40, which is also 2.5%, or you can use a graphic. They all represent the same thing, but for different people, one of those approaches can resonate more strongly than others. So if you have a BRCA1 or 2 mutation, what is your chances of developing cancer? Well, the risks range according to the study that has been done. And so follow me for a little bit here while I review these graphs. So this is a study that was done several years ago, which took all the existing studies that we had to date and said, what is the risk of breast cancer by a certain age in a BRCA1 or a BRCA2 mutation carrier? And each of the studies is a line, and the line length is a sense of how broad the confidence interval is, how certain we are. And you can see that for some studies, which are larger studies, the bar is shorter, and for some studies where the studies are smaller, the bar is a lot longer. And what we have in here in the black is sort of an estimate from those, a combination of all those studies. And you can see that the risk goes up, but you can also see these wide inter-study variations, all right? And you can see that for breast cancer, we're here, for BRCA1 and for BRCA2, and for ovarian cancer, the risk of ovarian cancer in BRCA2 carriers, you can even see in this curve, it's later and lower than in BRCA1. So this just gives you a sense of study by study how this impacts things. So in general, we give information in ranges. And depending on where you go and who you talk to, you'll probably get a slightly different range. But the general idea is that for women with BRCA1 mutations, the breast cancer risk is about 60 to 80%, ovarian cancer risk in the 30 to 45% range. There's a slight increased risk of pancreatic cancer. For BRCA2, also an increased risk of breast and ovarian cancer, although the ovarian cancer risk is somewhat lower. And there's a risk of pancreatic cancer and melanoma. And this gives the average risk, the risk to the average woman in the United States, just for reference. For men, as you can see, the risks for BRCA1 and BRCA2 mutations are listed here, including breast cancer, an increased risk of prostate cancer in BRCA2 carriers. This prostate cancer is generally more aggressive and higher grade. And there's an increased risk of pancreatic cancer, although for BRCA1, this is a very small increased risk. We also like to present risk by age. When people obtain this information that they have a BRCA1 or 2 mutation, they frequently view it as all their risk is in the present. It's all at them right this moment. But as you can see from this diagram, the risk, even for BRCA1 carriers, the risk of breast cancer by age 30 is very small. This risk goes up so that by age 40, it's in the 10% range, and you can see that by decade, we continue to increase this risk. But it does give you some sense that when someone might be tested at 25 or 26, that over the next 5 years, the risk of breast cancer is actually going to be quite low. Not zero, unfortunately, but low. Similarly, for BRCA2, you can see that by decade, the risk is low by 30, goes up by 40, and goes from there. And here are the BRCA1 specific ovarian cancer risks. This is important because as people try to make their decisions on when to remove their ovaries, you can see that the risk of developing an ovarian cancer by age 30 is extraordinarily small, and even by 40 is quite small. And so people really are making this decision usually for BRCA1 in this range because of these risk estimates. For BRCA2, these risks are even lower at these ages, and this is why oftentimes we talk about oophorectomy more sort of 40 to 45-ish in this particular group. The devil's in the details. A lot of individual factors apply. But the age-specific risks we find very important in counseling. So you're getting a sense of why risks are presented as ranges because the various studies have demonstrated a range, but why are there ranges in the first place? Well, it's because in addition to having this BRCA1 or 2 mutation, There are different types of BRCA1 and 2 mutations. We'll talk about that. There can be variations in other genes that can impact risk. There's other host factors like mammographic density or obesity, things like this. There's, in the different studies, different ascertained populations. By that I mean that in certain populations, the people that were enrolled in the studies all had very, very significant family histories of breast cancers, and others less so, and were more population-based studies, and that will impact risk. And we do know, although we don't know how to integrate these data into our existing models, that environmental exposures such as smoking and oral contraceptive pills can impact risk as well, although these are not dominant that we know about them right now for BRCA1 and 2 mutation carriers, and likely have a much greater impact in the general population. So why do I so often read that my risk of breast cancer is 87%? Well, this curve in yellow was the first study that really looked at the lifetime risk of breast cancer in BRCA1 and 2 mutation carriers. And I'll first point out that that was in 1995. So we've known about this information for 20 years, which is, not that long, and we've made quite a bit of progress since. But that very first study was the study in families that were used to find the BRCA1 and 2 mutations. And for that reason, these were very, very highly affected families. In order to enter this study, families had to have 4 or more living individuals with breast or ovarian cancer that could provide DNA samples for the research to go looking for these genes. Because this was very highly affected family members, it's potentially no surprise that the lifetime risk of breast cancer was 87% in this study. But in this same group, in BRCA1 mutation carriers, you can see that in other studies, the risks are much lower. This study here, by Fodor et al., back in 1998, examined consecutive breast cancer cases in a Jewish population in the Washington, D.C. area. And in their study, they found that the risk of breast cancer was more like in the 38% range. So you can see that this didn't select for family history, and this did, and this is the wide range that you can see. So family history and other potential genetic and environmental factors impact the risk of breast cancer. But these can be hard to tease out because families share other genes, families share environment, and families share the specific BRCA1 and 2 mutation. So we've been doing a lot of work on this area to try to figure out how to make sense of it all. So some of the early studies done 6 or 7 years ago looked at specific variation in other genes and said, okay, if you have these 2 other genes, and these are genes called SNP, single nucleotide polymorphisms, which only have a slight impact individually on them, on breast cancer risk. But if you have the higher risk copies of those and you have a BRCA2 mutation, it can impact risk. So let me show you that in more detail. In green, we have the average population risk of breast cancer. In the average population, if you also then have 2 of these other little genes and you have both bad copies, your risk goes up. Here it's seen in yellow. If you don't have either bad copy, your risk is in red, slightly lower than average. But if you apply that to a BRCA2 carrier where the risk is already quite high, this intervening line here, if you now have the bad copies, the risk goes up significantly compared to the average. And if you have both the good copies, it goes down a little bit. So the issue here is that the answer is high and really high. And whether people will do anything different with this information, we really don't know about yet. And again, this was early and wasn't validated. And so we've done a lot more work to try to get at that. And this is another example. This is BRCA1 mutation-associated risk. Ignore the black lines. They're just the outliers. But the black is the average risk. And in red is the group that has is in the 5th percentile of having high-risk variations in 10 other genes. So these are not BRCA1. These are other genes. And you can see that the curves separate to the higher and the lower risk groups. So I hope that's clear because it's a little confusing. But this is all-comer BRCA1 in this study. But the ones who had variation in other genes, you could tell either could be higher, or if they did not have that variation, they could be lower. And the same was true for ovarian cancer risk. So I've been asked by some people, then, send this test. I want to know it right now. And the answer is we're working on this. It had to be validated. There's a study coming out soon that really does show that this can potentially be impactful, but we don't know how to combine it with other information, as I'll show you. So we're not doing this in the clinic yet, but lots of folks are very interested in figuring out how we could use it. So this is coming soon, but not yet in a theater near you. So what about the specific mutations that we might have in BRCA1 or BRCA2? Does that matter? Well, it turns out that it might matter a little. So there had been small studies done before that had suggested that mutation specific mattered. And there was also a lot of interest about type of mutation. So let me explain this in more detail. Here you have a cartoon of the BRCA1 gene, and here's BRCA2. Both of these are very large genes with a large number of nucleotides. And they have different domains in them, different areas of the protein that do different things. It turns out that if there are mutations in specific areas, there appears to be a little bit more ovarian cancer risk than if there is a mutation in other genes. The challenge clinically is that these risk differences aren't very large, but it had been preliminarily seen. In addition, there was a lot of interest in whether a type of a mutation mattered. So you can have different types of mutations that do different things. One, and the most common type, is that a mutation takes a sentence and chops it off at the first word. That's called a truncating mutation. And those truncating mutations make a short little protein that then gets gobbled up by the cell. We call that nonsense-mediated decay. And some mutations are just missense mutations where a single letter is changed in this whole thing. Only a single letter is changed. So the protein will be made, and the question is whether it works or not. So all of this may go into whether people have a higher or lower risk of breast cancer depending on the exact type of mutation. So just to demonstrate that researchers really do sometimes collaborate with each other, and in fact, I think that the genetics field has been a real example of this and how this can be done. There is a consortium called the Consortium to Identify Modifiers of BRCA1 and 2. It's called SIMBA. And it's an international consortium of 55 sites from 33 countries on 6 continents that collect data on, and samples from BRCA1 and 2 mutation carriers. And the entry criteria for the study are pretty simple, which is that an individual just has to have a type of mutation in BRCA1 or 2. And for this particular data that I'm going to show you, there were more than 19,000 BRCA1 mutation carriers and almost 12,000 BRCA2 mutation carriers. So this is a really a huge testament to the patients who are willing to enroll in these studies, as well as a huge collaborative research project to try to get answers. So this, again, is this large study, and you can see that many women had breast cancer and ovarian cancer, but some people did not. They had neither type of cancer. And most women were white. We are actively working on this. We do not have a wide span of underrepresented minorities included in a lot of these studies, and it's extraordinarily important that we do better with that so that we have an appreciation for variation across the population. As I mentioned before, and I'm not going to get into this slide too much, except to say that depending on where the mutation is in the protein or in the DNA, it has differential impact on what happens with the protein. If you remember from high school or college or postgraduate biology, you have exons separated by what we call introns or intervening sequences, another exon, etc. And when you go through the process of making this into a protein, these areas are chopped up and made into mRNA without these intervening sequences. And you have a stop codon that's right here. The stop codon or you have a stop codon that's normally here, but if the mutation has made it so that the stop codon is earlier, it depends exactly where it is, whether or not it's going to mess things up or not. If the stop codon is too early, all of this protein will dissolve. If the stop codon is here, the protein will be made. The question will be whether it works or not. So we have to not only understand position, we also have to understand the biology. So for this study, we looked at breast and ovarian cancer. We looked at the mutation location, where in the gene the location was, and we also looked at the mutation type, which is this issue of whether the protein went away or not. And we performed these penetrance calculations, which is basically what is the risk of someone developing a breast or ovarian cancer. And as you can see, this is quite a detailed analysis. But the important thing is this slide, which I'm going to explain, which is in the central region of BRCA1, mutations in this region have a relative increased risk of ovarian cancer compared to mutations on the two ends. And this is not to say that the areas on the two ends in pink don't have an ovarian cancer risk. It's just that it is slightly higher in this risk in the middle, okay? Likewise, the mutations on the ends have a slightly higher risk of breast cancer than the risk of breast cancer related to the mutations in the middle. These are relatively small effects, but the important thing for scientists is not only can we use this information to help take care of patients if we can integrate it all together, but a potentially more interesting thing is why is it that certain mutations cause problems, and if we can understand that better, perhaps we can do a better job with our prevention approaches. So this is another way to look at the data, which is the types of mutations and what's associated with risk. And suffice it to say that it really summarizes what I've already just mentioned, which is that it depends on where it is. So the mutations in the middle have a relatively higher risk of ovarian cancer and a relatively lower risk of breast cancer, but these effect sizes are quite modest, and again, not to be used right now to make major decisions. A very similar thing is seen in BRCA2, which is the middle of the protein, seems relatively more ovarian cancer than the two ends. And depending on how you do your risk modeling in terms of what your lifetime risk of breast cancer might be, whether you use mutation-specific penetrance in light blue, looking at the specific genes, the specific mutations and what the risk is, or if you use more modeling approaches, you see that you can get somewhat different numbers. And again, will this be important? How are we going to use this information? We're going to have to see. So we'd also like to refine our risk calculations because we know that we need to incorporate many different things, but we would like to use this information about mutation location in cancer risk. And again, the mutations in the central region of both genes are associated with a relatively higher risk of ovarian cancer. And that can happen both due to increased risk of ovarian cancer because of those mutations or increased risk of breast cancer with the other mutations. The mutations associated with a continued presence of protein seem to be associated with a relative higher risk of breast cancer, but we don't have a good biologic explanation for that yet, and we'll be working on it. And again, this is only a first step to developing a more individualized model for people, and we're really going to have to do a lot more work on it. In order to do a lot more work, it's helpful to have the incorporation of as many BRCA1 and 2 mutation carriers as we can. And so here's information about the BASR registry, but again, wherever you live, if there's a registry nearby, please join. These are longitudinal studies. It helps us understand cancer risk, risk reduction, and risk modifiers, and serve as recruitment for other studies. And it's actually pretty straightforward. You can just do all this through the mail if you're interested, and this is the information. We have other registry studies done as well for mutations in other genes beyond BRCA1 and 2, and called the PROMPT study. And so these are ways that you can help the researchers get more information as quickly as we can. We are really trying to provide individualized risk information, but we're not exactly there yet. There's a lot of different resources that we have available on our website about cancer risk and genetics referrals, including areas near you and other things. So please check out our website at basser.org. And I did want to point out that a lot of the work that I just presented is coming from a lot of different people, and they're listed here, both at Penn and at many other groups, including this very large group of individuals that helped work on the piece of information for BRCA1 and 2. All right, so I'm going to start to take some questions because there are questions there. And the first question that I'll go to is, I am a BRCA1 ovarian cancer survivor in 2013. What is the standard of care related to mastectomy versus screening? And this is actually a really good question, and we do actually have information on this specifically. There have been 3 studies now done in BRCA1 and 2 mutation carriers with ovarian cancer, looking at their risk of breast cancer over the next 5 to 10 years. And in the first 5 years after an ovarian cancer diagnosis, the risk of breast cancer is actually relatively low. So the disease-free survival for breast cancer in the first 5 years after having an ovarian cancer is 95%. The other way to say that, there's about a 5% risk over 5 years that you'll develop breast cancer. In addition, those first 5 years after being diagnosed with ovarian cancer, the major risk is actually the risk of ovarian cancer recurring. And so we often like to just screen people for those first 5 years to make sure that the ovarian cancer isn't a problem in that time frame, and given the relatively low risk. So we generally do things like mammogram and MRI, but we put off the mastectomy for a little bit, just also to help people heal up and whatnot. Mastectomy is always a choice, not a requirement, but it always has to be factored in to what else is going on with you with your health, competing health. I have another question about models like the Gale model. So the Gale model is a great question. The Gale model is something that primary care physicians usually often use in their offices to estimate breast cancer risk. The issue with the Gale model is that it is absolutely useless for individuals who have a specific genetic susceptibility. So to say that again, if you have a BRCA1 mutation, the Gale model will dramatically underestimate your risk of developing breast cancer. The Gale model is really only useful in the general population. In the general population, without a strong family history of breast and ovarian cancer, and with with no suspicion that there's a high penetrance gene mutation, then the Gale model can be useful to figure out which individuals should have what we call chemoprevention, take medications to reduce the risk of breast cancer. But if you have a BRCA1 mutation, don't let your primary care doctor use the Gale model. All right, so the next question, you guys got good questions. I've heard that each generation of BRCA mutation carriers is getting breast and ovarian cancer much earlier than the prior generation. To what extent is this true, and how should I assess my risk by age in light of this? So one of the challenges with the data that we have in this light is the fact that we're doing so much more screening, so we may be impacting somewhat the age of diagnosis. We're also doing a lot more genetic testing. So there's a lot of pieces here that make things somewhat more challenging. Having said that, there is some evidence for genetic anticipation that cancers may come a little earlier, but this is not dramatically earlier, and it still does not change the recommendation that we in general start breast cancer screening at age 25. The only time we really go younger than that is if there's a really dramatically early breast cancer, sort of, in the, if a sister was diagnosed at 25. But even then, we at max go a couple of years younger than that. So we haven't really impacted that much at all. And there doesn't seem to be as much genetic anticipation for ovarian cancer risk. So this seems to be mostly a breast cancer risk phenomenon. The next question is the hope, as we better understand the impact of location on mutation risk, that we'll be able to change when individuals are suggested to begin screening or have prophylactic surgery? Yes, so we get so caught up in the definitions of statistical terms that you're exactly right. There's two ways that this information potentially could be used by patients, by individuals who are having to make decisions. The one is that individuals may decide, my risk isn't high enough that I want a mastectomy, I'm going to choose to screen. And that may be valid. But I think that, I'm not sure. I think that about that, because I think that people often make decisions about mastectomy for a lot of reasons, including their feeling about sexuality, quality of life, their personal history of cancer, where they are in their lives in terms of marriage and kids. There are so many elements of that. And so although the mutation-specific risk may play a role, again, we're probably talking about changing those ranges from 40 to 80%. So we're never getting down super low. But I do think that this information can definitely be used for timing. If we can validate this, and we can show that we're really accurate at predicting that someone's at a very low risk, for instance, of developing ovarian cancer by age 50, wouldn't it be great if we could wait till 50 to prophylactically remove the ovaries and spare someone all the downsides of that surgery? So I think that the timing of surgery could be really impacted by this type of information. And again, for some women, just the knowledge of risk may impact their decision making. But we actually do have a study plan to see exactly how people will use this information once we're able to give it to them accurately. So again, right now we're still a little hesitant on the accuracy of these mutation-specific risks and integrating these other cancer, other genes along with it. So we really envision this integrated model where we take your family history, the exact mutation you have, these single nucleotide polymorphisms and a bunch of other genes, environmental and reproductive factors, and try to give you a better risk estimate. A question came in about if you're overseas and you want to be part of the study. Sure, contact us and we'll figure that out. All right, another question was, we know that we had a big oncology conference last week, and what were some of the highlights related to BRCA1 and 2? So one of the big areas that we continue to work on is therapies for BRCA1 and 2 mutation carriers and what specific therapies we should use. There are drugs that continue to be in development called PARP inhibitors that have been studied in individuals with advanced cancer. So a study that we ran looking at pancreatic cancer in BRCA1 and 2 carriers showed that a PARP inhibitor called rucaparib didn't help as much as we wanted to help, but there were responses seen. There were studies done with PARP inhibitors in ovarian cancer and breast cancer. But what we're looking, so we're waiting for several very big studies related to that, which we'll be reporting soon. As a reminder, olaparib, which is a PARP inhibitor, is approved for BRCA1 and 2 related ovarian cancer that has come back, and for a woman who's received 3 types of chemotherapy already. But it is likely that those drugs will be approved for more things by the end of the year, we hope. We are interested in combining drugs to treat cancers better as well. There is a clinical trial for women with early stage breast cancer who have BRCA1 and 2 mutations after they're done with all their therapy, like their chemotherapy, to receive a PARP inhibitor called olaparib versus a placebo. And then there was a lot of work in the conference reported on things like the fact that underrepresented minorities, aren't getting as much testing and aren't doing as much with that information in terms of surgical procedures. So we have a ways to go of just taking the information that we currently know about and getting it out in the community so that individuals know about the opportunity to get tested, and then use that information to improve their medical care. All right. Let's see. There's some, okay. Yeah, someone asked a great question. I am a BRCA1 carrier, and I have a family history where people had stomach cancer, breast cancer, melanoma, and my cousin had breast cancer, but she tested negative for the BRCA mutation. So breast cancer is extremely common in the United States, particularly, and so for that reason, if in a family where there's, a bunch of women and cousins and aunts and things like that, it definitely can happen that someone can get breast cancer in the family that doesn't have the gene mutation in the family. And we even have a word for that in the genetics clinic. We call that a phenocopy. They don't, that individual, the cousin, doesn't have the same genotype. She doesn't have the same gene mutation, but she has the same phenotype, which is that she has a breast cancer. And that's what we call that. And so for breast cancer, it's not that uncommon that we see it. But for things like ovarian cancer, it's much, much less common. And this is why we always emphasize that genetic testing is just a piece of information. This is particularly true when you have someone who has a strong family history and tests negative for BRCA1 and 2 mutations. They still have a strong family history of breast cancer. They still need to be managed at elevated risk. So that's one thing. The other question is what other cancers might be associated with BRCA1 mutations. We don't usually think about melanoma. We think about that more with BRCA2. And the challenge of melanoma is that there is a lot of environmental exposure that can impact that. And stomach cancer is hard because this was your great-great-grandmother or great-grandmother, and sometimes that information is not accurate, and we always wonder whether that could be an ovarian cancer. So people are really quite good if they say that if their relative has had breast cancer. When we get pathology reports on that, it's almost always breast cancer. But when it's a different type of cancer, and particularly stomach cancer is the one that's really classic. Stomach cancer is tough, and it's often different types of cancer. So we feel strongly that people should know about their family history, document their family history, and let everyone else know about it so that as technology moves on and we have a better understanding of things, we'll be also able to give more specific advice. So, any other questions? It looks like I got most of them on my screen, but I'm happy to take others as well. And if not, thank you for joining us. We really appreciate it. We will be starting another webinar series next year as well. If there are specific topics that you're interested in, let us know that as well. Pam, do you have any other comments?
Just that the webinar will be archived for viewing. It'll be at basser.org/webinar. And our next webinar, like Dr. Domchek said, will be in the fall, and we'll post more information about that on our website and Facebook page. Just a final note, thank you again to Dr. Edward and Merry Prostic for making this event possible. We hope you enjoy the rest of your day.